Right, ladies and gentlemen, I think we'll get started. Thank you all very much for coming. This is very much the sort of graveyard shift of Groundswell, I suppose, isn't it? The very last talk on the last day. My name's Tom Allen Stevens. I'm the founder of British On-Farm Innovation Network, which I'm going to tell you a little about in a moment. And I've also got Tom Thirkell here from Crop Science Centre, and he's going to be presenting the really interesting stuff a bit later on. But I'd like to start by asking all of you just a little bit of a demonstration, first of all. Anyone who has been involved in on-farm trials, can you all stand up, please? Anyone who has been involved in on-farm trials, would you like to stand up, please? That's brilliant. Look at this. This is brilliant. Fantastic. Okay, anyone... No, don't sit down. You must stay standing. Okay, and now I'd like you guys stay standing, and I'd like those people who have ever had a spray miss and thought, "Ooh, that's a little bit interesting." Would you like to stand up as well? Anyone who has had any trials on their farm, would you like to stand up? Fantastic, wonderful. That's great. And if you've got an interest in on-farm trials, would you like to stand up? Join those people standing. Fantastic. Look at this. This is brilliant. And if you're just a little bit curious, stand up. Fantastic. Oh, look at this. Brilliant. Yep. So give yourselves a round of applause. We have a room full of on-farm innovators here. You should all join BOFIN. So what is BOFIN? Well, it's the British On-Farm Innovation Network. We've been going for about 3 years. We've got just over 500 members now, and it's to support people who do on-farm trials. And we've got a particular interest in new tech, bringing new tech onto farm and innovative things. So, what is farmer-led research? I always say it's for the farmer with the curious mind. You know, the sort of person who looks at a spray miss and goes, "Oh, that's a little bit interesting. What happened there, I wonder?" What we're trying to do is bring a bit more of the science into it. So a lot of people may do on-farm tramline trials, for example, and they look back and they think, "Oh, that did a bit better than that." But this is about putting some science, some figures behind it, and actually having something robust, resilient, that's repeatable on other farms. And we're looking to push the boundaries of science. I mean, this is a really exciting time to be in agriculture, I think. There are some really, really interesting innovations coming through, both within agriculture and in agri-tech and so on. What we're looking to do is push the boundaries of the fourth agricultural revolution. The other thing about this is that we've got a real cultural sharing. So this is about sharing and collaborating. And getting together, sharing experiences. You guys, you know it. You know, when you get into a room with a bunch of farmers and you start talking about it and sharing experiences, you know, it's brilliant. That is knowledge. That is the transfer of knowledge, and it's a very, very powerful and empowering thing. And so we want to generate that on a larger scale. That's our aim. So, and the other aim we've got is for farmers to be paid for on-farm trials. I'm going to tell you a little bit about some projects in a minute where we're doing that. So this is one project that we started a few years ago. I came across some researchers at Rothamsted who are doing some work. And I heard about some feeding trials that were going on at the John Innes Centre into a wheat, Watkins 788. It's an old landrace variety, hasn't been grown for 100 years, never been grown in the UK, came from Turkmenistan. And they found at John Innes Centre that in these slug feeding trials that it was that slugs consistently spurned it, but they didn't know for sure until they brought it out into the field. I said, this is perfect for an on-farm trial. And they said, "Oh, farmers won't want to grow this. It'll flop all over the place. It won't yield." So I put I only had 150 members at the time. I put the thought to them, and 30 of them came back and said, "Yep, really, really want to do this trial. When can we have the seed?" So I went back to the researchers, and they said, "Oh, we've only got 42 grams of seed." So anyway, on the strength of that, they multiplied it up. We had enough for a trial, and I can see one or two of our farmers in here actually who are doing the trial. it was when we started off, we didn't have any funding, but the wonderful, wonderful Environment Agency came forward and gave us some funding, which paid for the science. It also paid for some more on-farm trials. Some more feeding trials as well in the lab. So really exciting results coming through from that. I'm not sure I've got time to tell you, but perhaps you can ask me about it later. So these are the slug slews, and you'll see there are one or two of them in the audience today as well. One of the things that we do is, so we turn the scientific protocol, we work with scientists to turn the protocol into something that is manageable for farmers. You know, you're all time poor, you don't have too much time to do things. We give you as much assistance as we can to make sure that you can capture the data, that it's valid scientific data, but doesn't tie you to these plots. We're also working with Husk Data, who create this app that's really, really good at capturing this information. You can even capture photographs as well. So really good results. Well, I say really good results. Unfortunately, there were no slugs. Spoiler alert, we still don't know whether this wheat is actually resistant. But we've got another year, which I'll tell you about in a minute. But what we did gather is that the standard of the work done, bearing in mind this was farmers with no proper experience in on-farm trials, was up to the publishable standards you would expect from a professional trials technician. So isn't that fantastic? This is proper science. If anyone tells you that you can't do science in an on-farm trial, that is balderdash. It quite obviously can be. So the other element of this is that the way we delivered the trials was at about a third of the cost that it would cost for research staff to do the same work. So that's a real resounding thumbs up for on-farm research. Anyway, we're going to year 2 of this. The wonderful Innovate UK are funding this trial, and we're basically going to be repeating of what we did in the first year. We've got some more feeding trials going on, as well as the on-farm trials. What was interesting in the feeding trials, by the way, is that so the farmers said they not only wanted to look at the Watkins in the feeding trials, but also some of the varieties that they're actually growing on farm as the farm standard. And the interesting thing here is that, okay, so the Watkins performed as consistently as before, but the interesting thing is that one or two of the varieties, the farm standard varieties, perform pretty well as well. Not as well as the Watkins, but there is a spread. And this is something that is really, really interesting. So it's not as if, you know, all wheats are the same to slugs. They do differ. Gleam and Graham, for example, perform particularly well, so did X-Days. Dorsum, the slugs quite fancy Dorsum. But anyway, this is something, these are just preliminary results. You know, stay tuned and you'll hear more. So yeah, and then we've now launched. We launched yesterday a brand new project, Slimers: Strategies Leading to Improved Resilience, Improved Management and Enhanced Resilience Against Slugs. So this is a 2.6 million program. These are the partners that we've got involved in this. Really, really interesting one. I'm not going to dwell too much on this, but I've got information on it if you'd like to talk to me about it afterwards. But what I will say is we are looking for people to be involved in on-farm trials. We will pay you to do this work. It's a 3-year project. We've got some really exciting stuff going on here. Now, just bear in mind that slugs cost the industry £43.5 million a year, and we have just one form of control, ferric phosphate. I mean, what happens if we lose that? You know, what happens if there's another neonics or something? You know, we're stuffed. So this is about creating some more strategies to do it. As I say, I'm not going to go into too much detail, but I'd love to talk to you about it afterwards. Also, the NCS project. So this is a large industry consortium. We've got 17 different partners there. And this is about getting more pulses onto farms. So raising the amount of pulses grown to 20%, reducing the amount of soya fed in livestock rations by 50%. How are we going to do that? Well, it's simple. Just grow one and feed it into the other and knock out the soya. And the carbon savings are huge. 1.5 million tonnes per year. That's more than 50% that UK agriculture can actually achieve. And we can do that with your help. It's a farmer-led project. We're looking for 200 farmers to be involved. There will be farmers will be paid to do on-farm trials at the centre of this, and we're looking to build a farming community around it called the Pulse PEP. So we'd like you to join the Pulse PEP, be involved in the project, and and shape how it comes through. So another one, just quickly, harvest weed seed control. So this is, you may have heard about these seed control units that go on the back of the combine, extensively used in Canada and Australia. Not really used very much in Europe or the UK, but that's what we're looking to assess. So we know that the seed that passes through it and goes through the chaff stream and it gets mashed up by these units, about 98% control. What we don't know is actually what's going in the front of the combine, how much seed is still standing at harvest. So we're working with NIAB, that Will Smith, who's been doing this, and he's developed a protocol for measuring this, and we're now rolling it out. We've got about 50 seed scouts involved who are going to be measuring this at harvest. So we'll have a really nice data set by harvest of just what this can actually perform. Again, it's another Innovate UK funded program. We did the initial trials last year, and we got a second year coming up. And you can see the details there. Again, what we do is we create this protocol, and we try to make instructions that are as clear as possible to follow. This one's actually led by Adam Driver, who many of you may know. It's a farmer-led project. I have to say, is there anyone here who's dealt with Innovate UK? Yeah. Do you like dealing with Innovate UK? No, I mean they're lovely funders. Don't get me wrong. Most of my income comes from Innovate UK, so I've got to be a little bit careful here. But yeah, it's a nightmare system to deal with. But anyway, I'm not going to dwell on that. This is the one that you all want to hear about, that you're here to talk about. I'm not going to say too much about this, apart from. So I came across Tom because he's been doing some really interesting research in gene editing, which is my thing. I just love it. And it was through that we got talking about mycorrhizal fungi, and then this project developed. When Tom first brought it to me, I thought, yes, this is absolutely what BOFIN members want to do. And sure enough, you came through, and we had a really, really good uptake from this. So thank you very much for all of you who got involved and joined our soil circle. And then we have at the center of that the people who actually did the work and collected the samples and sent them in as well. I'm not going to say too much about that, obviously, because you'll hear more about it from someone who's far more qualified than I am. So I'll hand over to Tom now to give the results.
Okay, thank you very much. Thank you, Tom. Okay, so yeah, making the most of mycorrhizas and what the Soil Squad found. So very briefly, I'll just say where I've come from. I'm a researcher at the Crop Science Centre, and we are an institute staffed by members of the University of Cambridge and NIAB, and it's all about fostering translational research and trying to get stuff from lab discoveries in the greenhouse out into the field and testing in realistic environments. So, what are we talking about today? The Soil Squad are looking for arbuscular mycorrhizal fungi, and I'm sure a lot of you know this is a type of soil fungi. They're growing through your soils, and they also grow into the roots of plants, and this lets them form this association, this nutrient exchange association. So the fungi are scavenging phosphorus and nitrogen, other nutrients and water from the soils, passing them onto the plants, and then in return the plants are providing them with carbon. And as well as improving nutrient uptake, they can also help a plant with its defense responses, so they can respond quicker and more effectively against pests and pathogens. They're improving plant water use efficiency, and they can also help with a plant's heavy metal tolerance, and sort of a more wider scale, soils that are rich in these fungi have got often show improved soil structure and even reduced greenhouse gas emissions. And the two little photos on the bottom left there, you can see the leftmost one is the soil hyphae, filaments growing through the soil, and then to the right, all that in dark blue is selective staining of mycorrhizal tissue inside the roots. And it's often said that plants don't have roots, they have mycorrhizas. And this is because very few plants take up nutrients exclusively through their roots. About 90% of plants use mycorrhizal fungi of some type or other to get their nutrients. About 70 or 80% use arbuscular mycorrhizas. All of the top 10 crops in the world use the arbuscular mycorrhizal symbiosis for improved nutrient uptake. And because all the plants use them, that means they're in all the soils. The plants are supporting them. And so you've got this population, this community of these fungi in all the soils. We're still not entirely sure everything of what they're doing and the situations in which they're doing them, hence this project and my research more generally. And they're enormously abundant. So a kilogram of soil can contain 30 kilometers of arbuscular mycorrhizal fungal hyphae. And we know there are at least 200 species that we've found and described so far. There's bound to be many more. And for their importance to plant nutrient uptake, we know that they can, in some circumstances, contribute 100% of plant phosphorus uptake. And they're very abundant, but they've still, because of these nutritional effects, got a sort of extra. They've got more of an effect than you'd expect them to have. And so they're often described as ecosystem engineers. They're a really keystone, important part of the soil microbiome. And so some big questions we still really need to get to the details of. What are mycorrhizas doing in agricultural systems? What are agricultural practices doing to the mycorrhizas? And then how can we use and optimize those mycorrhizas in agriculture? What are they doing in agricultural systems? Sometimes a lot. We've got a study here. I wasn't involved in this research. This was a group in Mexico who have gene edited some maize plants. And on the left, you've got AMF susceptible, so these are your wild-type normal maize plants that can freely form this association. The gene-edited line is the AMF resistant, so it can't form this association, so it can't get those benefits. And they did really badly. You can hopefully see they're much yellower, they're more stunted, the cobs were much smaller on these maize plants, and the authors found that they were contributing 30% of the yield to this maize. Moving to potatoes, and this is an inoculation trial. This was a meta-analysis of 231 studies where they inoculated potatoes, and they showed that consistently these mycorrhizas are boosting potato yield, so you've got a 10% increase in yield across this. Having said that, one was in maize, one was in potatoes. It would be great to have this type of analysis in all crops and all soils, and I can tell you, go ahead and all this, but it's still difficult to draw general trends. So please don't ask me what should I do on my soil with my crops, because I'm still not sure. Hence our BOFIN project. What are agricultural practices doing to mycorrhizas? In general, speaking very generally, intensive practices are bad for mycorrhizas, and the more you do, the worse it is. This study here from a group in Switzerland, I wasn't involved in it, but I wish I had been, because it's really incredibly, it's so robust and impressive, and they sampled, I think, from 130 farms and just did a lot. So I'm going to talk you through some of the things they show. What we've got here is fungicides reduce mycorrhizal phosphorus uptake. Mycorrhizal fungi are fungi, fungicides are bad for fungi. From left to right on this graph, you've got the number of fungicide applications that go onto a soil. And then up and down, so the height of those dots is the amount of phosphorus those fungi are providing to the plants. And so the bars are getting shorter. You can see that the more fungicide you apply, the less benefit you're getting from those fungi in the soil. Perhaps not surprising, but what might surprise you is that this sort of level of data is very rare, because it takes so long to get. It's really difficult to get this sort of scale of data. Phosphorus fertilizer. Along the bottom, from left to right, you've got your Olsen P, your phosphorus content of your soils, and again, you've got the mycorrhizal contribution to those plants. If your soil phosphorus is high, the mycorrhizas are doing very little for your plants. And then tillage, so soil disturbance, mechanical disturbance of your soil. We know that it's really bad for the abundance and the diversity of these really fine fungal filaments. They're everywhere, but if you keep turning the soil over and smashing it up, they're not going to do well. And again, the height of the dots on the graph are the benefit they're giving to the plants in terms of their phosphorus. And unfortunately, if you've got a system with low mycorrhizal diversity, so your number of species in your soil, you're on the left-hand side of this graph, and you're getting the lowest benefit from those fungi. So it's not only the amount of fungi you have in there, but it's the number of fungal species as well that get you your benefit. So what am I going to say? Disturbance is really bad. Don't disturb your soils in any way, and that's great for the mycorrhizas, but that's not going to give you much of a crop at the end of the year. So some disturbance is obviously required. So looking at how to optimize mycorrhizas in agriculture, I think you can't just do it with one fix. This is a big, complex problem, and I think it needs the right genes in the right plants in the right soils. So I'm going to quickly walk you through this. You don't need to know much of this detail. Basically, at the Crop Science Centre and wider groups of collaborators, we've got a really good idea of which genes control mycorrhizal colonization in plants. And the two bars on the left show you the amount of mycorrhizas in your roots in a normal wild-type barley plant when you've got low soil phosphorus. They're really high. If you put loads of phosphorus on your soils, the plants don't need the mycorrhizas because the phosphorus is just there available. And so your amount of mycorrhizal colonization goes almost to zero. And what we found is that we can control this with genetics. So we've got this NSP2 overexpressor. We've got a GM plant here. The communication between the fungus and the plant has been enhanced. And so the plants are recruiting more of these fungi. And now you should see the two bars on the right-hand side, you've got increased mycorrhizas. So in this GM line, that high phosphorus is no longer suppressing the mycorrhizal colonization. And we're hopeful that if we can get more mycorrhizas into the roots, that means they can deliver more benefits to our plants. The right plants. So another complication. Not all plant genotypes respond equally to these fungi. What I've got here is 8 wheat lines from a mapping population, so they're incredibly closely related, these lines of wheat. And I inoculated with the same fungi, and in those 4 lines on the left, the fungi decreased the plant growth. And for reasons I don't understand, the 4 lines on the right, the fungi had a hugely beneficial effect. And so next steps on this is to dig into the genetics of this wheat and see why some respond really positively and some respond really negatively. Because, as I say, these fungi are everywhere in our soils. We're managing the soils to keep the diversity and the abundance high. If we then put plants in that respond negatively to that abundance and diversity, we're working against ourselves. So put crops that are going to benefit into the soils and should be good. So this is largely recovering what I've already said, but putting those right genes in the right plants in the right soils. We're looking for abundant and diverse mycorrhizal networks, which are probably usually most beneficial to our plants. We're trying to reduce to a minimum the chemical, physical, and biological disturbances of these systems. We know that the mycorrhizas are in those soils. We don't really know how beneficial they always are. And something I almost touched on just on the last slide was looking for this sort of Goldilocks zone of disturbance where we can maintain the benefit of these fungi but also practically get a crop off the field as well. So, first steps was a sampling survey with BOFIN. We're looking to see how much mycorrhizal fungus is in UK farm soils, how different management practices affect this, and are the fungi the same by location and management. And we have to start small and sensible, so we asked people to sample the same crop, which was winter wheat, at a similar time, just before flowering, or certainly before flowering. That's when you're getting peak nutrient uptake and when the plants are going to be relying most on these fungi, and to a standard protocol. So we wrote a set of instructions and sent that out to our farmers. And we had about 70 farmers initially register, and then I think we sent out about 50 packs to farmers who, once they realised what we were asking them to do, still wanted to take part. And so we sent out a sampling pack, asked them to follow the instructions and then send their roots back. And I was a bit embarrassed to tell Tom this, but I was amazed. Every single pack came back exactly following the instructions. The tubes were all filled as we'd asked. All fantastic. It was brilliant. As Tom says, it really shows the power of this sort of network to do this rigorous research. And so sample analysis, which is still ongoing, I'm using selective staining to quantify the mycorrhizal fungi and then visualize the structures in those roots. Samples that are in the freezer now, I'm going to do some DNA sequencing on that, and we can tell which fungal species are in our different soils, in our wheat roots. And I'm going to show you very soon the first lot of results, but we've got much more to come. So these next couple of slides are an incomplete picture, but they're showing some nice trends. So hopefully you can read the text at the bottom of this graph, but from left to right we have our different cropping systems, and up and down the text has disappeared, but the height of the bars represents the amount of fungus that's in these roots. And so far left you can see from the soils that were managed conventionally, ploughed, these have got the lowest, so about 10% of the root system has got mycorrhizal fungus in it. And then moving through, increasing the amount of colonisation. We've got our regenerative agriculture with shallow non-inversion tilling, got increased up to 25%. Regenerative agriculture plus direct drill, a little bit higher still. And then our highest was the organics. And the pictures left and right, the one on the left you can see wheat root, and hopefully you can see that all of that blue colour in the middle is mycorrhizal fungal tissue. They really can dominate the root system. So when people say plants don't have roots, they have mycorrhizas, this is another aspect of that the fungal biomass can be really surprisingly high in these roots. And on the right, it's not come out so well, but you can hopefully see it's again a wheat root that's been stained, and you've got those fungal hyphae spreading out away from the roots. And I've dunked these in ethanol, in potassium hydroxide, in stains. So some has clung on, most of it obviously has broken off and is still in your soils, but I was happy to find some there still growing out. And then this is a similar graph showing a very similar trend, the arbuscule. So this is what the fungi are named after. It's the structure inside the root that allows them to exchange nutrients. And yes, it's good to know how much fungi in total is in there, but the arbuscules really give us a clue about the amount of nutrient exchange that's happening. And similarly, we get the lowest in our conventional and ploughed system. Perhaps not surprising. Regen plus shallow non-inversion, regen and direct drill, slight difference there perhaps, and then clearly organic much higher. So it's suggesting to us that our mycorrhizas in the organic system are not only more abundant, but they're probably doing more for their plants as well. So that was a very quick little intro to the project, much more data to come, so watch this space. And I'd like to say thank you all for listening, and thank you to all of our funders, especially RAGT and KWS, who supported this project. And thank you.
Thank you, Tom. Can we change back? I have to say, that's the first time I've seen the results. Tom wouldn't even tell me what they were. So this is absolutely hot off the press. And I have to say, I think the really fascinating... Sorry, is the microphone working? Yes, it is. A really fascinating results there. So thank you very much, Tom, for your work. I think it was quite painstaking work went into that as well. So really good news. So I just wanted to finish off by just having a look at where this might be going. So I just want to tell you a little bit about the way we do things at BOFIN. So at the centre of a trial will be the actual trialist farmers. In this instance, the soil squad who are doing the work. But the way we work is that we involve people in what's called the soil circle, or it's a knowledge cluster that we build around a project. And this is a really important part of it. So there are some people, some people who really want to do the trial work, but not everyone. Doesn't suit everyone. Or perhaps they're not farmers, but they've got some specialist knowledge. They've got some interest in it. And so we welcome everyone into this soil circle where we can discuss things. We can help shape the project. We can bring expert knowledge in, and so on. And this is how we run all of our projects. It's not just about the trial itself. It's about the discussion around it. And that's really important because that's knowledge, and knowledge has value. And all of you who contribute that knowledge, all of those ideas, that has value. And you should get the value from that knowledge. That's what we're trying to do. And that also provides the bridge to what I call the innovation community. So that would include the early adopters, the first people who would take up the technology once it comes out into the commercial marketplace. And it's about building that discussion, building the ideas around it, and the knowledge sharing is what it's all about. And we know that there's no better way of learning about a new practice than hearing it from another farmer, and that's what it's all about. It's about farmer to farmer learning. That's what we're trying to generate. And as I say, you get really, really great discussions when you sit down and you talk things through. So let's try and get more of that. Not just face to face, but online as well. And we're looking to build platforms that enable that discussion. So we've come up with this idea. So we're looking for funding for this idea, but basically, so it's the thing. It's about the truth about your soils. So thriving roots underpinning total soil health. Yeah, there's a stray S in there. I couldn't get them. Anyway, so this is what we're trying to work on now, getting a project underway, so, myself as a farmer, I sat down, what do I actually want to know about what's going on beneath my feet? I know so much about the plant growth above it, and we've got so many different ways of measuring it. We're told so many things. So we've actually become quite good at the breeding of this stuff above the soil. We haven't got a clue what's going on below the soil, really. We can't see it, we can't measure it, and so on. So I want to know what the optimum root architecture is as well, and I want to know how that is interacting with the soil microbes, and this is what we've been doing with the AMF project. But then also, how do you manage the soil root pathogens? And it's really interesting, that work that you were talking about, with that there is some pathogenic activity that goes on there as well. We need to understand those interactions. And then there's the interventions that we put in, and well, the first one is not really an intervention; it's a way of measuring it. How can we have a way of measuring what's going on? But then, how do genetics play into that? And so we know so much about the genetics above the ground now. We know very, very little about what's going on with the genetics below the ground. But, that's another intervention that we can play in here. And then there's, biostimulants. There's so much of these biostimulants and biologicals that are going on here. And I don't know, just how much science is there behind these things? Do we actually know? Have we got a way of measuring it? Have us farmers got a way of measuring it? A platform, if you like, that we can do it. And then, of course, there's what influence cultivations, companion cropping, and all those sorts of things have on it. And we've just seen a snapshot of that. But there's there's a lot of work that goes into this. But just how much scientific understanding have we got? So we've come up to this idea of the matrix of truth. Sorry, I should have mentioned that we've been working with CHAP on this idea, developing it. So the matrix of truth, and you've got the sort of fundamental questions on the left-hand side there, and then you've got the interventions along the top. What we want to do is, have a series of work packages that look at these various interventions, and then some on-farm trials that really look at, what the interactions are between them. So this is what we want to do, and these are the outcomes. I mean, the main thing that we want to establish is a platform, a farmer-led platform where we can actually measure this. And there are ways of measuring what's going on beneath your soil, but it's quite laborious. It involves quite a lot of interventions and so on. There's not really an awful lot of science that you can actually practice on farm at the moment. So we want to get that, and we want to get as proxy measurements as well, so that you don't have to do too much interventions, but you can get a good idea of what's going on through some proxy measurements. And then we want to look at the genetics. There's some really exciting work going on now, looking at genetics and bringing in some of these landraised varieties, what we can learn from that and feed into new varieties. And then, so if we can deliver a suite of on-farm practices and target parameters that can be adapted to optimize soil health and crop productivity, this is about creating a platform that we can use as farmers to actually assess for ourselves, for our knowledge about what's going on beneath the soil and the interactions there. That's what the aim is. We're not there yet. We're looking to look for funding for this. We're building together partners into a collaborative project, but we're hoping that we can bring something forward. But that's the aspiration. We can build in some of Tom's work as well into this whole picture. So that's about it from me. Here are my details. Please do join BOFIN. Keep abreast of what's going on. Now, I believe the So the results, so those people, thank you very much, by the way, to all of those people who sent in samples and did the work. It was quite a lot of work for those people who did it, but I mean, what a testament to on-farm science, that every single sample was perfectly presented, perfectly packaged, with a full set of crop history data as well. So if you're one of those farmers, give yourselves a really big pat on the back, because you've done such a lot for science. Yeah, absolutely. And I think, so what's happening, the next step, Tom, do you want to just...
Yeah, it'll be continuing to characterize the colonization and DNA sequencing to identify the communities in the rest of the samples. I think I've done less than half of them so far. And it's also looking at these different trends and patterns. So the first thing that sprung to mind was to look at the soil management of these systems, and which, broad classes of management, but we got a lot of detail, as Tom says, from the questionnaire that we included. So previous crops on that soil type, location around the country. So there's lots more to data mining. Once we've got the data in, we can then see what trends and patterns there are from all of that. So yeah, that's to come.
If anyone's got some questions, then please put your hand up. But just before you do, I'm just going to ask Tom, so the individual farmers who have contributed, when will they hear their results?
Oh, that's a good question. You're going to make me say it. Soon. Soon. They will hear soon. Yeah.
I know what you're
putting together a results package for everyone that's contributed to the project, so they get some photos and data and a little bit of information and stuff in there as well.
And they'll get their own tailored results then,
yes, absolutely. Yeah. And we're not sharing them, so you're not being identified unless you want to be. Share amongst yourselves, but I'm not going to publish who's got what.
I might say John Cherry was, I think he sent his sample in, didn't he? Yes, he did. And also Andy Cato sent in a sample as well. So obviously we won't be identifying where they come on the scale, but they will know. Anyway, so are there any questions from the audience? We've got one here.
On the organic farm and the results from them, what tillage practices were used?
Off the top of my head, I don't remember. I believe they were direct draw, but I can get back to you on that. Sorry, I'll very quickly say, it's my intention with this to make it more publicly available. So whether that's through an academic publication or through BOFIN, so that we get a full report, and then all of this sort of stuff can go into the details, so people can see for themselves all the details of what's going on.
I mean, that's very much what BOFIN does in terms of delivering the results. So Tom will do his academic paper for all those highbrow scientists and so on, but we then turn it into something that's a bit more easy read, farmer friendly stuff that we'll be relaying to our members. So there was a question over here, I think.
Brilliant, thank you for the talk. It was really interesting to see the results. As I'm sure you know, mycorrhiza express lots of different plastic variations in their behavior. So as you saw, the saprotrophs versus the pathogens versus the symbiotrophs, and how they can change between those behaviors depending on the kind of circumstances. And I was wondering if you were looking at all into the functional profile of the behaviors of these systems rather than by species, because it might reveal more about what actually is going on below ground rather than what individual species may do when they may change their behavior because they've come into contact with some specific kind of chemical or environmental variable.
Yeah, that's absolutely something I want to do, is to pick out a number of these soils, isolate the fungi from them, use them, and all of this stuff is complicated and tricky to work out. So this is a bit that the next bit you can't do easily on farm. So you want really controlled conditions for working out the function, what those fungi are doing individually, if that makes sense. So you control the environment and the growth medium and stuff, and then you inoculate the plants with those, and then you can look at nutrient exchange studies. So you're using radioactive isotopes and all sorts to track where phosphorus moves through the system. And then maybe a lot of farmers won't want radioactive phosphorus dosed around their farms. But yeah, absolutely, looking at what they're doing as well as who they are. Yeah.
Great. Any other questions? Yes, a guy in the orange jacket at the back there.
Having given up ploughing about 50 years ago, I find it very frustrating to hear it ploughing referred to as conventional. Wonder how long we've got to go on not doing it. And I really think you ought to quite right to describe ploughing. And actually, if you think back years before the metal moldboard plough, a wooden plough didn't actually invert soil. So please don't talk about ploughing as conventional.
Noted. Thank you.
Well, just to clarify there, so we asked the farmers two things. So first of all, what their tillage system was, whether it was ploughed or min-till or direct drill, and then also how they describe their farming system. Are you conventional, are you regenerative, or are you organic? So we're certainly not saying that ploughing is conventional, but having said that, I would imagine that there were very few regenerative farmers who were ploughing, because that kind of goes against the grain. So so when Tom was talking about conventional ploughing, that was two different questions that we asked them.
Thank you.
Any other? Guy in the orange jacket there.
Hi Tom, sorry, I'm going to ask you a question not on your work, but on one of the papers you talked about. So the AM resistant kind of, I can't remember what the crop was, where you show 30% improvement. Maize.
Yep.
30% improvement. I've always wondered with that when you're doing genetic engineering, how you control for just other, because the gene knockouts are never, they might not be perfect, it might have other knock-on effects. So how do they know that that's entirely down to their kind of mycorrhiza and not something else?
That's a very good question. So there's a number of different controls you can do to test that. When you're doing gene editing, you knock out one gene, that's your target. And what about other genes that you might have accidentally knocked out? Well, the CRISPR gene editing technique is very precise, so it's very rare for it to knock stuff out, but you can then go back and do whole genome resequencing, and you can check that the other genes are all still there and you've not accidentally knocked out two genes that may interact. I guess I was curious if it have other effects, though. So not just say you hit the right gene, but then that can have other effects.
Could you bring it?
Sorry, that can have, instead of yelling. I guess I'm curious as well, though, if you knock out the right one, you evidence that by kind of going back and genetically sequencing it. But then what if that has other effects kind of further on that single gene? Because that can have...
Yeah, it may well do. And to answer your question initially, to control for that, it's very difficult to. So you're kind of assuming there that you're not, by knocking out that one gene, also affecting other processes. That gene may do things other than interact or change the way the plant interacts with mycorrhizal fungi. But yeah, that is a challenge of that approach. But where your plants are susceptible to mycorrhizas and your soils have got the inoculum in it, if you want to know the effect of mycorrhizas out in the soil, in the real world, your options are incredibly limited. There was an approach used, which is not used anymore, thankfully, is that the soils would be drenched in fungicide to destroy all of the mycorrhizal fungi, plus a lot of other things that are in there. Then you would grow plants there, plus a untreated soil, and say, there's the difference. So there's no perfect way of testing the role of mycorrhizas in the field, but I think gene editing is at least as good as the others.
Any other? Yes, at the front here.
Hello. I'm curious about the method that you used to inoculate the grain that you experimented on.
For the 8 genotypes I used.
For the wheat, when you compared on the table that some of the wheat varieties don't respond to mycorrhizal positive. And
yep. We used a pure culture of mycorrhizal spores, and it's from a lab at the University of Leuven. I forget the name of the lab, but they have a spin-out company that produces mycorrhizal spores. And so we grew the plants in a sterilized sand and perlite mix, so there's no other microbes initially in there. And then we grow them in the glass house either with these mycorrhizal spores or not. And then we compare the growth of those plants. So it's as tightly controlled as we can, and only changing whether or not there's mycorrhizal fungi in there. And obviously I know wheat doesn't normally grow in sand and perlite, but yeah.
We had another question over here, yep.
You said that you'd had soils sprayed with fungicides that had killed the mycorrhiza.
Yep.
Were spraying crops, spraying plants, is that going to have any effect on the mycorrhiza? I'm spraying my crops with a fungicide in the growth stages, because I'm always told that the fungicides move up, not down.
Yes, yeah, I've heard that as well. I'm no expert on the movement of fungicides, but I've seen studies, and a lot of data is suggesting that it definitely does have an impact. But as I sort of touched on before, you've got to get a crop at the end of the year, and so if you've got to use a fungicide to control a mildew or a rust or something, then so be it. And then the mycorrhizas will recover, and it's not like adding a fungicide is going to kill all the mycorrhizas off in your soil. It might knock them back a little bit, but they're still there, so they're going to recover. But I guess you're going to be wanting to use enough fungicide and no more anyway, so I guess that shouldn't be disastrous, but
definitely handled there. Any other questions? Yes, at the back there.
So are you actually in a position with these different wheats that you tested and found different results that you've got no way to distinguish them? That you can't point to any reason, like in their pedigree, how you group them?
No.
So I have read that like heritage wheats seem on average per wheat to have better mycorrhizal associations, and quite a lot of modern are bad at it, basically.
Yep. Yeah. These trends are definitely there, and they're coming out, and so it's definitely true. So the really old land races and stuff seem to associate more with these mycorrhizal fungi than the modern wheats, and I don't know how much that's because the modern varieties are bred for a more intensive system than the land races would have been. What impact that's having, I'm not sure. You know, if the mycorrhiza is still beneficial or not.
Can I just follow up to that? Because partly I'm looking at like the results, say, from the Rothamsted Broadbank, where they found that the older wheats had, post-Green Revolution, there's less micronutrient per grain, and that isn't just the question of grains per square metre. So is it possible, and I'm kind of hypothesising that when mycorrhizal associations are formed in order to get for the plant to find macronutrients, more or less automatically, as a byproduct, more micronutrients are also available or get into the grain?
Yes, short answer, yes. There's good data showing that mycorrhizas can also help grasses take up zinc and boron, potassium, maybe not a micronutrient, but certainly not just phosphorus and nitrogen.
Can we leave that to flavour? There seems to be some evidence. I mean, I've seen one talk here of people saying, we can actually tell in our mouth how much micronutrients there is in the food, and we like that food where it's got lots.
I don't know about that.
I think did we have any landrace varieties in the samples that were sent in?
I'm not sure.
No.
I'll check. I don't think so, but maybe.
Most of them were Skyfall, RGT Skyfall and KWS X-Days were most of the samples. We did try to keep it quite restrictive, but I mean that would be a really interesting place to take this work, perhaps. Look at whether the landrace varieties do something different, and whether there's something a trait that we can locate and perhaps bring into conventional, elite varieties that would make more of that association. That would be really interesting. Sorry, any other questions? There's one here, and
Thank you. It's more of an observation than a question, but as I understand it, plants evolve. They have co-evolved with the mycorrhizal associations. So that's maybe why older varieties have stronger associations. We've co-evolved with microbial associations. So if I understand it right, is this trial based on looking for more mycorrhizal associations to improve yield? Yeah. So maybe you also need to look at the flip side, and look at mycorrhiza as well, not just those not just narrow it down into the particular strains that you've got and that you found, and look for more variety out in the world that already exists.
Yeah.
Yeah, I think, and also one other thing, it's a system that relies on other microbial associations, and it seems to me quite odd to then isolate this one aspect of a system. That's just my where I come from.
Yep. Yeah, no, that all very good points. But it's the mycorrhizas we know have a oversized effect. Their effect is really substantial, and the soil microbial community is hideously complex. And then looking at environmental variations on that, cropping variations, in terms of working out how we can get a benefit from those, we have to isolate things. So what we're doing here with mycorrhizas is starting with one that we know is really important. But as you've suggested, we know that the mycorrhizas are interacting with bacteria. There are bacteria that solubilize phosphorus, which makes it available for the mycorrhizal fungi to collect and then to pass it to the plant. So fixing mycorrhizas on their own, I absolutely agree, is not going to sort everything. So yeah, lots of complex and interacting things to look at.
I mean, the main aim of this project was to get an understanding of just what the range was of mycorrhizals across UK soils and across farming systems and so on, and just get a picture of just how much it does vary. Because as far as I gather, I think this is the first time this work has actually been done, that we've actually had a national survey like this.
I think so. I think Farah may have done something similar, but certainly the first sort of farmer participation survey like this.
Yeah. So I mean, obviously there are all sorts of ways in which we can develop this work, you know, and this is just the sort of thing, you know, and where we take this, it should be farmer-led, and so, you know, and if you've got particular thoughts about where it should go, then please do join the soil circle, join the conversation, and let's see where that goes. Anyway, there was another question over here somewhere. Yes.
Just to, we're watching machines working outside there. Have you looked at all at transference of mycorrhiza from one crop to the next in any shape or form? Because part of the sequence of cover cropping and whatever is you're putting as one crop is passing on or dying out or becoming mature, you're planting seeds in for the next crop. And there's quite a bit of anecdotal information about transference of mycorrhiza. Have you looked at any of that at all?
I haven't personally, but I know that it's been observed that, keeping roots alive in the soil for as long as you can is providing carbon for these fungi to maintain that mycelium, that body of filaments in the soil. And so, yes, you're going from one crop in the rotation to the next. That first crop is sustaining that mycorrhizal community to then infect the next, to colonize the next. So yeah, absolutely. And if you have a year in your rotation with non-mycorrhizal plants, so brassicas or sugar beet or something, then they're not supporting that mycorrhizal community. They're still there because they're going to retreat into spores, and they'll sort of equivalent of overwintering, but however long, until you have a plant that can host them again. And then that will just pick up, and then that community is supported again.
Got a question down here. Sorry, wait for the microphone.
Would companion cropping help?
Yes, I think it definitely would.
If you had canola and then combine it with a...
Yeah, because I've sampled from a field of oilseed rape, which is a non-host for mycorrhizal fungi. I sampled some grass weeds in that field, and they were all colonized by mycorrhizal fungi. So they're there and they're active. They're just waiting for a plant that is a suitable host for a root of one of those plants to come by. The spores will germinate, and then they'll colonize those roots. Yeah.
Great. So my black grass is doing a job in the oilseed rape. That's wonderful. Any other questions? We got any other? No, I think that's. We've got another question here.
Hi again. Regarding the last question, Isn't that mean that if we have perennial woody plants, they will be the ultimate companion that will keep the mycorrhizal thriving until the next crop?
If they are a suitable host, yes. There's a lot of woody plants that are hosts for arbuscular mycorrhizas. There's a number of different classes of mycorrhizal fungal groupings, depending on their different fungal families and their different. They grow differently, slightly differently. Pine trees and oak trees and things and beeches use a different group of fungi, which you can have them on a field margin, but they're not going to colonize your crops because they don't interact with these sorts of plants. But yes, if you've got a sort of a reserve, like a hedgerow or something, the mycorrhizas are going to forage and explore out of there, and they'll find those plants that are good hosts. So, yeah, and there's a project, I can't remember who led it, but it may be University of Sheffield or Leeds, where they're investigating exactly this sort of question, the role of hedgerows in fungi and bacteria migrating back into a crop field, the importance of hedgerows in that sort of system. Yeah.
I guess that would also count for agroforestry as well.
Absolutely. Yeah.
Yes, another question here.
So I'm just thinking back to one of your slides where regen, non-inversion tillage had a higher percentage than regen no-till. Is that enough of an increase to sort of justify the negatives that come with tillage?
Talking as a mycorrhizal biologist, I would say maybe. No, I don't think so. I think you have to look much more broader. I can't advise you on what you should do based on that graph, because that would be silly, because you know your system so much. I don't know yours at all. So I've had the roots that you've sent in, and I can tell you what mycorrhizal fungi are there. So, no, don't know. I think the important thing to take away from that data are the broad trends, that more mechanical soil disturbance was bad for the fungi and less was good. So.
Yeah, okay, thank you.
We've got another question at the back there.
Really, this is going back to one of your slides about the phosphate usage and way phosphate doesn't like, or mitochondria doesn't like phosphate. We're paid by or obliged by Southern Water to put on sewage sludge. I guess that's becoming a complete no-no under what you're suggesting. What's your view on that sort of application? Because it's been a good fertilizer.
Yeah. Again, talking from somebody whose career is researching mycorrhizas, I'd say, oh, that's a shame that you're impairing your mycorrhizal abundance and diversity and function in there. But the mycorrhizas aren't always the only route that your plants are going to get nutrients. So ultimately, it's going to be about doing the best in terms of getting a crop, and then what the water company's saying about pollution. And yeah, so that's a non-answer, I'm afraid. But
no, well, that's right. I think we've really, literally, just scratched the surface here, and there's so much more to understand, to explore beneath our soils. And as I say, this research should be farmer-led. It's our knowledge, it's our systems, and we should benefit from that knowledge. We should have the value from it. Thank you very much, everybody, for coming. Thank you very much to Tom for presenting the results. It's been fascinating working with Tom and exploring this. And as I say, it's just the start. And I hope you'll all join us as we carry on in this work. As I say, we are very much looking to develop this into other projects, other areas. And as I say, it should be farmer-led. So please do guide us as to where we go. But great. No, thank you very much. I'd just like to ask you to put your hands together for Tom. Thank you.
All right, for Tom. Thank you.